Awesome, thank you. I know I'm the last one before lunch, so 5 quick minutes. I only have 56 slides, so it should be no problem at all. So my name is Brad. I'm here from the Bioinformatics Core at Harvard School of Public Health, and I'm going to talk quick 5 minutes about work we're doing to try and prioritize structural variants. And so a lot of what we do is build community-developed tools to take advantage of existing resources, so put together tools into a framework that is validated and can work nicely together. And so we specifically have a tool called BOSC NextGen, which is kind of not a black box, a clear box that takes samples and configuration, and you can see all the insides, and it runs a bunch of tools, scales them, and gives you on the output stuff like variations in RNA-seq, quality control, validation, feeding into open source analysis tools, things like that. And we've been doing more work recently on cancer. And one thing that's very important in cancer is that structural variant changes drive a lot of the important biology that's happening. So this is a nice paper on pancreatic cancer, which describes, among other things, the impact of structural variants. And you don't really know much other than like all the boxes mean there's a structural variant there, and you can see that a lot of these cancers are defined by structural variants, and these ones on this side are labeled unstable because there's crazy chromotripsis happening all over the place. So it's clearly key to be able to identify these. The difficult part is technically it's really hard to do. This is a validation of different structural variant callers. And you can sort of see the sensitivity and precision. Used to, like, if you're doing SNPs and indels, used to 99% precision or 90% precision, and sensitivity. Here you're talking about 70%, 60% sensitivity and 50% precision. So half the things you look at are going to be fake. So you really have a lot more than you can look at, and you're not sure about them, and you need to be able to drill in on them. And so we need a way to take that big mess of things where there's important data in there and pull out what that important data is. And so the really simple-minded approach we're taking is to try to prioritize these based on known biological information. So you can bin the genome into any way you want to bin the genome. You can make a bed file, typically transcript, something like that. And then you take a set of known variations and you build a priority database, which is a fancy name for a big bed file that's gzipped and has information in it. And then you can take your raw calls and run it through a set of tools and get prioritized calls in their side. So you're basically pulling out the things that should be obvious, that if you were, say, to look through every single one by hand and you were a cancer expert, you would highlight those. The idea is to build a tool that will do that for you. And so to do this, you need good quality databases with cancer variants, things that you would pull out if you were a cancer expert. And so there's some cool tools available, like Civic. Malachi is actually talking about that tomorrow, so I'm excited about that. Integen. But these are sort of tools. These are databases in development. If you just sit down and read a textbook, 900-page textbook about the biology of cancer, there's a lot of information that we know that you could put in your brain that you're not able to compute on because there's not a computable structure for it. So, I think one of the challenges for us is not only in the software side, but also in organizing this data and putting it together. So my quick summary of that is structure variants are critical for cancer and a lot of other things as well. The cell doesn't care how it's rearranging stuff. It's only just because it's harder to do, for us to detect structure variants doesn't mean they're less biologically important, but they're hard to do. So we can use known biological information to prioritize the important things. And I think one of the key components from reading about this, learning about this, is we really need to support community-based databases that underlie all this technology, that let us take our knowledge and put it into practice. And so if this is interesting to you, this code is all available. I have a little tool called BOSC Prioritize, which does the little mess around with bed files and things like that. And then all of this is work we're doing in conjunction with BOSC Bio, which is the larger framework, does all the variant calling and validation and those kind of things. And I know it's lunch, so I'm happy to talk to people afterwards. If you have questions or anything about that, I'll be around all days. So awesome, lunchtime.